us look at this word soma. Now, somatic embryogenesis. Now, soma means body cell. Okay, what does it mean? It is a non-sexual cell. Alright, so it can be a cell from a leaf, it can be cells or tissues from the stem, it can be from the seeds, alright, it can be even from the pollen grains, or it can even be from the new cells. When you look at this process, development of embryo, you need a male plant, you need a female plant, so it is a sexual process. Okay, and you need two sexual units in the form of microspore and in the form of the egg. But when you look at somatic embryogenesis, okay, it is a non-sexual process. You don't need a male plant, you don't need a female plant, but rather any cell, it can be the cell or the tissues from the leaf, from the stem, from the seed or from the nucellus, these can give rise to embryo. Alright, these can give rise to embryo. Okay, look here. Now Stewart, he was the person, alright, Stewart, in the year 1956, alright, he was the first person who reported the phenomenon of somatic embryogenesis in carrots. Okay, he was the first person who reported the development of embryo from carrots. So you all know what is carrot. Example, this is carrot. Okay, if this is a carrot, alright, so carrot is like this. He took the carrot, what did he do? He cut thin section of the carrots. This is carrot. So your carrot is your starting material, explant. Alright, he took the carrot and then made it into these thin slices, and they should contain this at the center. This at the center, when you cut carrots, when you slice carrot, you will see that at the center there is a tissue here which is orange in color. This tissue is actually the tracheary elements. What does it mean? It contains the xylem as well as it contains the phloem. And what are these? These are the vascular system of the plant. Alright, these are the vascular system of the plant. Okay, so he took this and then he cut the carrot into thin sections, and then what did he do next? He prepared a media. Okay, he prepared a media, and in this media it is auxin rich. This media is very rich in auxin. Alright, it contains a high amount of auxin, little cytokinin, less of cytokinin, more of auxin. It contains low amount of dissolved oxygen. Okay, then it contains nitrate, ammonium nitrate, which is a source of what? Which is a source of nitrogen. Alright, and then it also contains potassium ions. Okay, he prepared this media, and auxin rich media. Next, what did he do? He poured the media in the test tubes. Okay, he poured the media in the test tubes, and then he took the carrot slices and then he cultured it here in this auxin rich media, and these test tubes were cultured at a temperature of 18 to 30 degrees C. That is the temperature. Okay, and then in the presence of a high light intensity of about 1600 to 1400 lux. That is the light intensity. Then what did he found? He found found that after some time, what happened to these carrot slices after some time? A mass of cell developed from this. Okay, these slices, they disappear and instead a clump appear, and he called it the callus. Okay, now this callus contained thousands and hundreds of cells. Then what did he do next? Next, he took the test tube and then he poured media. In this media there is no oxygen. Remember, we started with a media which is oxygen rich. What did he do? The moment he got callus, he removed oxygen. The rest remained the same. That is, cytokinin is still there, low oxygen is still there, nitrogen is still there, and potassium ion source is still there. What happened to this callus? From this callus, he could see heart-shaped structures arising. Okay, from this callus, many heart-shaped structures arises. He took the structure and then he see under the microscope, analyze under the microscope. And when he analyzed under the microscope, he found that these structures are actually this, the heart-shaped embryo. Okay, it contains this, the bipolar axis, which is the axis that all embryos should have, okay, for the development of a complete plant. Alright, then he called these structures as embryoids instead of embryo, because these embryoids, they arises through a non-sexual process. He did not start, look here, he did not start with a male parent or a female parent. He started with a carrot. And what is a carrot? You all know a carrot is a root, a taproot. Okay, so what is this carrot? It is a body cell, it is a somatic cell, it is the body of a plant. What do you call that? A sporophyte. It is a sporophyte. And from this carrot, what did he do? Simply by culturing the species of carrot, okay, in an oxygen-rich media, he could induce the formation of a callus, and from a callus he could get embryos. Okay, and these embryos, alright, they contain the bipolar structure that all embryos should have. Alright, and this non-sexual process through which embryos can be given out, it is known as somatic embryogenesis. Later it has been found that not only carrot, even your cauliflower, okay, cauliflower plant, from this plant you can get embryos. Alright, from about 71 species of angiosperm, Stewart found out that he could induce them to produce embryoids. He do not have to wait for the male plant, he do not have to wait for the female plant. Alright, from the sporophytic body, from the somatic body, they could give rise to embryo-like structure. So this is somatic embryogenesis. This process, alright, it is non-traditional, because why? It takes place in the lab. And it does not need a male plant, it does not need a female plant. It just needs some inducing agents, alright? And those inducing agents can be in the form of your growth hormones, okay, oxygen, nitrogen, these mineral supplements in the form of potassium ion, and then you have to tweak, you have to play with temperature, you have to play with the light intensity. Okay, now this is one of the most important process that could replace micropropagation in future.